So lecanemab slows cognitive decline in Alzheimer's patients by 27%, which, I mean, as a monoclonal antibody targeting amyloid-beta, it's just a massive clinical breakthrough.
Oh, absolutely. It's huge.
Right. But if we are being completely honest about the pharmacology here, until this recent paper dropped in Nature Neuroscience, the field was essentially, flying blind regarding why it actually outperformed other anti-amyloid therapies.
Yes, that's exactly it.
Like, we had the clinical outcome, sure, but the actual intracellular mechanics were just this complete black box.
The data bears that out entirely, yeah.
Yeah.
Because for years, the prevailing assumption in neuropharmacology has been that these drugs act primarily as passive flags.
Passive flags.
Right. The idea was just that the antibody binds to the toxic protein and that simple act of binding is enough to, alert the immune system to come and clear it away.
Just tag it and wait.
Exactly.
Yeah.
But this new research proves the interaction is far more instructive and honestly far more complex than a simple tagging mechanism.
Okay, let's unpack this, because our mission for this deep dive today is to really look at the middlemen here.
The microglia.
Exactly. We are not just analyzing the drug, and we are not just looking at the amyloid plaques. We are dissecting how lecanemab fundamentally rewires the brain's resident immune cells, those microglia, and we are going all the way down to the transcriptional level to do it.
Which is such a critical shift in perspective. I mean, when we analyze Alzheimer's pathology, like through tissue samples or neuroimaging, we consistently observe this really strange paradox.
Right, the stalling out.
Yeah, we see microglia clustered super densely around the amyloid plaques. They are physically right there, adjacent to the toxic A-beta 42 aggregates, but they are in this stalled state.
We call them disease-associated microglia, right? Or DAM.
Exactly, DAM. They are metabolically active, they're trying to work, yet they just fail to effectively phagocytose or, ingest the amyloid.
So they basically mobilize to the site of the damage, like a bunch of janitors rushing to a spill, but then they essentially freeze.
That's a perfect analogy, yeah.
And studying how to kickstart those stalled cells presents a massive logistical hurdle. I mean, you obviously cannot run real-time single-cell transcriptomics on a living human brain while dosing them with an experimental drug.
No, definitely not.
But you also can't just use a standard laboratory mouse, because mouse microglia do not mirror the human transcriptomic response to amyloid pathology at all. They just operate on totally different biochemical pathways.
Which raises an incredibly important question, right? How do you test a human-specific immune response in a preclinical model?
Right. How do you bridge that gap?
Well, the researchers solved this by engineering this incredibly sophisticated transgenic environment. It's the NLGF CSF1R Delta Fire mouse model.
Okay, I want to break down exactly how this model works, because the bioengineering here is just fascinating.
It really is.
Let's start with the first half of that super long nomenclature, the NLGF, because they didn't just overexpress the amyloid precursor protein to force plaques to form, right?
No, because doing that can create artificial phenotypes. It's not natural.
Right. Instead, they inserted specific human Alzheimer's mutations, the Swedish, Iberian, and Arctic mutations, straight into the endogenous mouse gene.
Yes, which forces the mouse to naturally deposit these human-like, toxic A-beta 42 plaques at a totally physiological rate.
So the pathology is humanized, but the true innovation is in the second half, right? The CSF1R Delta Fire mutation.
Exactly. So the CF1R gene is absolutely essential for the survival and development of microglia. And what the researchers did was utilize CRISPR to delete a specific enhancer region within this gene.
This is the fire enhancer, right? The MM's intronic regulatory element.
You got it. And by deleting that single enhancer, it creates this highly specific biological void. The mouse develops entirely normally in, every other physiological respect.
But it's born completely lacking its own endogenous mouse microglia.
Exactly. It creates this totally empty ecological niche within the central nervous system.
So by starving the native microglial lineage of that crucial survival signal, the brain is prepped for engraftment.
Precisely. So then, the researchers transplanted human-induced pluripotent stem cell-derived microglia into the brains of these newborn mice.
Wow.
Yeah, and without any competition from native cells, the human microglia just populate the brain. They establish distinct territories and behave exactly as they would in a human neural environment.
Okay, so to synthesize what we're looking at before we even introduce the drug, let's do our first mini recap. So bullet 1 for you listening. We have engineered a living system where the brain produces human-like amyloid pathology.
Right.
Bullet 2. The actual immune response is driven entirely by transplanted human microglial cells. It's basically a biological sandbox.
I like that, a biological sandbox.
Yeah, and bullet 3. We have completely circumvented the cross-species translation gap. Because, if you've ever tracked a promising drug that cured Alzheimer's in a mouse but failed completely in human trials, this transcriptomic mismatch is usually exactly why.
Yeah, it's a huge bottleneck in the field.
Yeah.
But this humanized model provides the exact platform needed to finally test lecanemab's true mechanism of action.
Okay, so with the board set, the researchers could finally introduce the mystery drug. But to understand the experimental design here, we really have to look at the biomechanics of a monoclonal antibody.
Right, you have to picture the structure.
Yeah, so picture a standard IgG antibody as a Y-shaped protein complex. The two arms at the top, the Fab regions, those are the variable domains.
The grabbers, essentially.
Exactly. These highly customized grabbers that lock onto the specific target, which in this case is the specific conformation of the A-beta 42 plaque. But the stem of the Y, the Fc fragment, that's what actually dictates the biological response.
Right. The Fc fragment, or fragment crystallizable region, is not just some structural tether. It's a highly potent receptor agonist.
Okay, so what happens when it binds?
Well, once the Fab arms bind to the amyloid, that Fc stem binds to specific Fc gamma receptors located on the surface of the microglia. And it's this engagement that initiates these massive intracellular signaling cascades.
So to determine if that tail engagement was the actual driver of the plaque clearance, the researchers didn't just guess, right? They manipulated the variable.
Exactly. They engineered a mutated version of the drug known as LLA-PG lecanemab.
LLA-PG. What does that actually do to the drug?
So the LLA-PG mutation involves highly targeted amino acid substitutions in that Fc region. Specifically, they convert leucine to alanine at positions 234 and 235, and they substitute proline with glycine at position 329.
Basically acting like a mute button on the tail.
Exactly. Biochemically, this completely abrogates the tail's ability to bind to the microglial Fc gamma receptors. It totally neutralizes the effector function.
But wait, let me push back on the spatial mechanics here for a second. If you introduce these mutations and fundamentally alter the conformation of the Fc tail, doesn't that structural shift risk, compromising the Fab region at the top?
It's a valid concern.
Like, are we entirely sure the LAPG version can still effectively bind to the amyloid plaque in the first place? Because if it can't bind, the whole experiment is basically moot.
Right, and it is a vital control to establish. But confocal microscopy confirmed that the binding affinity of the Fab region remained entirely intact.
Oh, really?
Yeah, in fact, the LLA-PG antibody bound to the amyloid plaques perfectly. Over an 8-week observation period, the mutated drug just piled up heavily on the surface of the plaques.
But the tail was muted.
Exactly. Because the Evel-Hayes tail was silenced, the microglia completely failed to recognize it. The drug just accumulated passively.
So the microglia essentially ignored it despite the plaque being heavily coated in this expensive antibody.
They remained in that stalled, disease-associated state, yeah. But the contrast with the unmutated normal lecanemab was just stark.
What happened there?
Well, in the cohort treated with standard lecanemab, the drug did not accumulate on the plaque surface. Instead, immunofluorescence revealed the antibody was entirely internalized within the human microglia.
Wow, so they ate it.
Yes. The cells had recognized the active Fc tail, engulfed the entire plaque antibody complex, and actively cleared the insoluble A-beta 42.
That brings up a massive safety concern, though. I mean, when you aggressively stimulate microglial phagocytosis, when you trigger this literal feeding frenzy in the brain's immune system, the immediate clinical fear is synaptophagy, right?
Yes, absolutely.
Like, are these hyperactivated microglia going rogue and indiscriminately pruning healthy dendritic spines alongside the toxic amyloid?
And that off-target damage is historically one of the great pitfalls of neuroimmunotherapies.
So how did they check for that?
To evaluate the synaptic integrity, the researchers quantified 2 very specific protein markers. First, synaptophysin, which indicates the structural health of presynaptic vesicles, and second, Homer1, which is a marker for the postsynaptic density.
Both of which obviously need to remain stable if your neural circuits are going to keep functioning.
Exactly, and the data show they were completely preserved. The lecanemab-treated mice exhibited no synaptic loss whatsoever.
It's incredible.
It gets better. Furthermore, the researchers tracked LMP1, which is a biomarker that highlights dystrophic neurites, these swollen, damaged axonal branches that typically surround toxic plaques.
The collateral damage zones.
Right. And under standard lecanemab treatment, LMP1 levels decrease significantly. The microglial clearance was not only highly targeted, but it actually allowed the surrounding neural architecture to actively heal.
And just as a definitive proof of concept, when they administered lecanemab to a control group of mice that lacked microglia entirely, the drug accomplished absolutely nothing.
Nothing at all. The plaques remained entirely untouched. What's fascinating here is that the drug itself doesn't destroy the disease.
Right. Let's do our second mini recap to lock this in. So, tracking our progress through this mechanism, bullet 1, the drug binds the plaque. Bullet 2, the tail of the drug engages the microglia. The efficacy hinges entirely on this Fc receptor engagement. It is not a molecular bulldozer, it's a highly specific key. And bullet 3, with that engagement, the microglia safely eat the plaque without harming the synapses. The drug just provides the exact authorization code the body needs to do the work safely.
That's a great way to put it.
But this brings us to the core intracellular mystery, doesn't it? We know that the Fc tail forces the microglia to eat the plaque, but how does that simple receptor engagement fundamentally rewire the cell's internal behavior?
And to decode that intracellular signaling cascade, the researchers had to map the microglial transcriptome. They needed to see exactly which genes were upregulated and which were suppressed.
Here's where it gets really interesting, because historically doing this meant relying on bulk RNA sequencing, right?
Right. You would basically extract the tissue, homogenize it, and sequence the RNA.
The problem with that is that bulk sequencing provides an average expression across millions of cells, which completely obliterates the spatial context. You lose the ability to see how a cell's physical location influences its behavior. It's like trying to understand the dynamics of a major city by blending all the residents together into a smoothie and measuring their average stress levels. You wouldn't know if the stress was coming from the financial district or the quiet suburbs.
That is precisely the issue. So to solve this, the researchers utilized spatial transcriptomics, specifically a high-resolution platform called NovaST, and paired it with single-cell RNA sequencing.
And what did that show them?
Well, the level of resolution NovaST provides is just stunning. It allows us to map gene expression directly onto the histological landscape of the brain tissue.
So they could see the map?
Yes. The researchers could look at a specific microglial cell, measure its exact physical distance from an amyloid plaque in micrometers, and read its unique transcriptional profile simultaneously.
And the spatial data revealed a really clear gradient, didn't it? In the normal lecanemab cohort, as the microglia physically approached the plaque, an entirely distinct set of genes began to skyrocket.
Yes, specifically the pathways governing phagocytosis biogenesis and lysosomal acidification.
Basically the cellular machinery for ingestion and digestion.
Right. But the researchers needed to identify the master regulatory networks driving this massive shift. So to isolate these networks, they applied an advanced computational algorithm known as weighted gene co-expression network analysis, or WGCNA.
WGCNA is such an incredibly powerful tool for cutting through transcriptomic noise. It analyzes thousands of genes and groups them into these modules based on their co-expression patterns.
Correct.
Like if a specific cluster of genes always turns on and off together in response to a stimulus, WGCNA identifies them as a functional network and assigns them a color code.
And out of all the networks identified here, one specific co-expression cluster emerged as the definitive driver of lecanemab's efficacy, the pink module.
The pink module, it represents this highly specialized transcriptional state that is entirely decoupled from canonical neuroinflammation.
Yes, and when they analyzed the architecture of this pink module, they found one specific gene was anchoring the entire network.
The SPP1 gene.
Exactly, SPP1, which encodes a matricellular protein called osteopontin, or OPN.
OPN?
Yeah. Now, while OPN is known to play various roles in tissue remodeling and immune modulation, its aggressive upregulation in this specific spatial context, exclusively in microglia adjacent to plaques and exclusively in the presence of an active Fc tail, was totally unprecedented.
We really have to contrast this dynamic with earlier anti-amyloid therapies to truly understand why this SPP1 activation is so groundbreaking. Let's look at aducanumab for a second, which is an older monoclonal antibody.
Right, good comparison.
When aducanumab binds to amyloid and engages microglia, it does trigger clearance, But it activates an entirely different transcriptional network, doesn't it?
It does. WGCNA identified the aducanumab response as the Brown module. And the Brown module is characterized by aggressive canonical inflammatory markers.
The bad stuff.
Yeah, genes encoding tumor necrosis factor, or TNF, and interleukin-1 beta. It essentially triggers a severe cytokine storm localized entirely within the neural tissue.
And that biological scorched earth policy translates directly to the severe adverse events we see in clinical settings.
Exactly.
The overactivation of that Brown module is what drives ARIA, right? Amyloid-related imaging abnormalities. It causes microhemorrhages, vascular leakage, and this really dangerous edema in the brain.
It's a huge problem.
The immune system clears the plaques, sure, but it causes massive collateral damage in the process.
And that is the pivotal distinction here. Lecanemab actively suppressed that inflammatory Brown module. It bypassed the destructive pathways entirely and channeled the cellular energy almost exclusively into the pink module.
It initiated a targeted, non-inflammatory phagocytic response driven by osteopontin.
Exactly.
But, correlation does not equal causation. Just because SPP1 is upregulated doesn't inherently prove that osteopontin is the actual executioner of the plaque clearance. How did they prove that OPN was driving the physical breakdown of the amyloid?
They designed this incredibly elegant in vitro validation model. So they cultured slices of mouse brain tissue that were heavily burdened with human amyloid plaques.
Okay.
They then seeded these slices with human microglia. But instead of adding lecanemab to the environment, they introduced human recombinant osteopontin directly onto the tissue.
Wait, they removed the drug entirely and just provided the downstream protein?
Exactly. And the results were definitive. The introduction of exogenous OPN alone at the appropriate concentration was completely sufficient to trigger the microglia to engulf and clear the amyloid plaques.
Wow.
It confirmed absolutely that lecanemab's efficacy is ultimately reliant on its ability to flip the SBP1 genetic switch.
Let's pull this complex cascade together for our third and final mini recap. So we started with the stalled immune system. By utilizing high-resolution spatial transcriptomics, we can clearly see that intact lecanemab bypasses the destructive inflammatory networks that plague older drugs.
Right.
Instead, its XC tail acts as a precise transcriptional switch. It engages the receptor, upregulates the pink module, and drives the massive production of osteopontin.
Which is the SPP1 gene.
Exactly. And that single protein then transforms the microglia from passive bystanders into highly targeted, non-toxic plaque clearers. I mean, if you are tracking the evolution of neuropharmacology, this is a leap.
It is a massive leap.
We are no longer just designing drugs that bind to targets. We're designing therapies that dictate specific cellular behavior after the binding occurs. So what does this all mean?
Well, if we connect this to the bigger picture, it fundamentally changes how we approach drug development for neurodegenerative diseases as whole.
How so?
Because for decades, the metric for success was simple target engagement. Can we build a molecule sticky enough to attach to the pathology?
Massive flag.
Exactly. But this paper proves that binding is merely step one. The future of precision medicine lies in molecular programming. We need to engineer therapies that communicate effectively with the host's immune system, utilizing the nuanced language of specific receptor agonists to trigger precise, localized genetic responses.
All without initiating systemic inflammation. It really is a blueprint for the next generation of therapeutics. And not just in Alzheimer's, but potentially in Parkinson's, ALS, multiple sclerosis. We're learning how to hand the immune system a highly specific set of instructions rather than just sounding a generic alarm and hoping for the best.
It's an entirely new paradigm.
And that leads me to a final provocative thought I want to leave you with as we wrap up today's deep dive. Because this entire mechanism relies on machinery that already exists within the cell.
That's true.
The SPP1 gene and the capacity to produce osteopontin were always there, encoded in the microglia, just waiting for the precise sequence of receptor engagements to be unlocked.
Just waiting to be switched on.
Right. So if lecanemab works by unlocking a latent, dormant superpower inside our immune cells, what other dormant healing states are hiding in our biology? What other non-inflammatory, highly regenerative transcriptional modules are just waiting in the genome, requiring nothing more than the correct molecular key to wake them up?
That's a fascinating question.
The hardware to heal the brain might already be installed. We just have to crack the code. Thank you so much for joining us on this deep dive. Keep asking questions, keep looking past the binary, and keep exploring.